Yeah, welcome to my YouTube channel. Thanks very much for the subscriptions. For those who have subscribed, those ones who have not yet subscribed, please do one thing: subscribe, put a notification, and we'll continue to learn. Last time we saw one of our major and important pathways in the body as regards to cell proliferation, control of cell proliferation, and differentiation of different types of cells in the body. And that was the Hippo pathway, which was involved in size regulation of organs. Today we are going to be looking at JAK-STAT pathway. JAK-STAT pathway is involved in different processes in the body, such as immunity and the blood cell differentiation. We have hematopoiesis control and cell proliferation and the others. The JAK-STAT pathway has proteins. It has the JAK, which is the Janus kinase. Then we have the STAT, which is the signal transducer and activator of transcription. So the JAK proteins, they are 4 in number. We have from JAK1 to JAK3, then TYK, K2 proteins. Then the STAT has 7 proteins. There are 7 STAT proteins from STAT 1 to 6, then STAT 5A and 5B. The JAK1 protein is involved in interferon gamma activation. Then the JAK3 is involved in lymphocyte development. Then the STAT4 is involved in NK cell activation. Then STAT5 is involved in white blood cell differentiation. So when we go to the pathway, we are going to see that we have our cytokine receptor. This is our receptor, the cytokine receptor. We have our ligand in blue, it's the cell membrane and the proteins. So what happens? What happens in this pathway? When our ligand binds to our cytokine receptor, what is going to happen next? We are going to see activation of the receptor. When the receptor is activated, they are going to dimerize. So they exist as monomers. So once the ligand binds to the receptor, they are going to dimerize after being activated. So their dimerization attracts JAK proteins. JAK proteins come and bind on the cytosolic part of the dimerized receptor. After our JAK protein binding, what else? The JAK protein is going to do what we call transphosphorylation of the cytosolic part of the receptor. After transphosphorylation, this is the phosphorylation of this receptor is going to attract, is going to produce sites for binding for all those proteins which have an all the protein with SH2 domain. So one of the proteins with SH2 domain, we have the STAT protein. So we are seeing the STAT protein coming to bind to what? The receptor. After it binding here, it's going to be phosphorylated. So this is phosphorylation of the STAT protein. It's going to dimerize. After dimerization, the activated STAT protein is going to translocate into the nucleus. Inside the nucleus, it's going to trigger transcription of required target genes. So this SH2 domain acts as a link between the JAK-STAT protein, between the JAK-STAT pathway and other pathways, such as the tyrosine kinase and other pathways. In tyrosine kinase, we have proteins like mammalian target of rapamycin. We have others. So this SH2 domain is going to link this pathway to other pathways. The hallmark of cancer related to this pathway is self-sufficiency. What are the cancers that are? We have the leukemias, we have myelodysplastic syndrome, we have colorectal cancers, and thrombocytopenia. So those are some of the conditions that are going to arise due to mutation of the genes that are responsible for coding for these JAK and STAT proteins.